We're here at the Scripps Institution of Oceanography in La Jolla, California, in the laboratory of Dr. Michael Latz. And we're here to visit with him and to have him tell us a little bit about his research. So he's an expert in bioluminescence, and maybe you can tell us a little bit about the work that you do here in the lab.
Well, we study small unicellular plankton called dinoflagellates. The nice thing about our research is that we don't have to go out to the ocean and collect them for every experiment, but we can grow them here in the laboratory. So we raise them, grow them in flasks like this. And in here we have seawater, and we've added nutrients. And then we put a little bit of the dinoflagellates in, and then they grow. And they grow like plants. Dinoflagellates are not plants, but they are photosynthetic, and so they get their energy from light, and we grow them like houseplants. So the nice thing about this is that we can grow them to be very abundant, and then we use them in our experiments.
So tell us about your experiments with bioluminescence, the dinoflagellates. They perform bioluminescence, and maybe you can tell us some of the experiments that you do with them.
Well, one of the experiments we've been working on recently is understanding how a single cell responds to a stimulus. Now, in the case of these organisms, dinoflagellates, they sense a mechanical stimulus. So in the case of in the ocean, a predator grabs them and tries to eat them. And the force of the grabbing is what acts as the stimulus, and then they respond with a flash of light. And that flash of light has a purpose of trying to distract or scare off the predator. So the predator that's grabbed it sees the flash of light and gets startled, and then that affects the feeding behavior. So what we want to understand is how that stimulus of the being grabbed ends up as a flash of light.
Now they're clear here during the daytime, and of course the lights are on here. But are they bioluminescent now?
Well, they have a very strict day and night schedule of their bioluminescence, just like we have our own day and night cycles. So if I said to you, okay, go to sleep for 5 minutes and then wake up, well, your body just isn't on that kind of rhythm. Dinoflagellates have day and night rhythms just like we do. In fact, they use the same types of chemicals in their cell as our brain uses in our own bodies. So no, these dinoflagellates here think it's daytime, the lights are on, but it's only at nighttime when the bioluminescence system is turned on. So when we're studying their bioluminescence in the laboratory, we need to trick them in a way. We shift their day and night cycle so that in our afternoon, it is actually their nighttime, and then we can do experiments during our afternoon. So we just shift their cycle just like someone working the night shift gets used to that schedule, and they're fine with that, and it's a lot more convenient for us to do research during our afternoon than during our nighttime.
So tell us some of the experiments that you do with these dinoflagellates. We know they bioluminesce, but what exactly are you working on to try to get them to do?
Well, one of the things that we're interested in is how flow stimulates bioluminescence. Naturally, a predator grabs the cell, and that's the stimulus for them to flash. But if you've been on a beach with the waves breaking on a dark night and seen bioluminescence, you'll notice that the waves breaking stimulates the light, or fish swimming through the water stimulates the cells as well. So it's a mechanical stimulus that stimulates them, and we're very interested in the levels of the flow stimuli. So we do experiments in the laboratory where we take cells like this, but during their nighttime, and we subject them to flows that are very defined in terms of the physics. And then we can relate the physical conditions in the flows to the stimulation of bioluminescence, and then we begin to understand the flow conditions that stimulates them to bioluminesce.
Very interesting. Well, thanks for sharing with us some of your research that you do here at Scripps.
Thank you.